Welcome to the glycolysis cycle. This cycle happens in the cytoplasm of the cell, and in our energy investment phase, we're going to spin 2 ATP to break apart this glucose molecule. That's going to leave us with NAD+, 2 ATP, and 2 pyruvates. That is the energy investment phase. And then the NAD+ is going to oxidize into NADH and give us 2 more ATP. Acetyl coenzyme A is going to enter the mitochondrial matrix and join oxoacetylate, forming citrate to start the citric acid cycle. Now, enzymes are going to go ahead and rearrange these molecules, releasing CO2 and converting NAD+ to NADH and FAD to FADH2. Now, each one of these rotations that we have is going to make 1 ATP and give us CO2 and FADH2 and NADH so we can begin our next cycle. Glucose, and then that is a 6-carbon structure, and we're going to take one of those carbons actually, and we're going to release it as CO2, forming an exergonic reaction, and we're also going to oxidize the pyruvates, meaning that we are going to strip it of its electrons. And then we're going to go ahead and pop that on NADH, and we're going to make it NAD+. And this is all happening in the mitochondrial matrix. And then this now acetyl group is going to attach to coenzyme A to form acetyl coenzyme A. The electron transport chain, which is happening in the inner mitochondrial membrane. And then here we see ATP synthase, which are protein complexes, and then they pump protons across and in the inner mitochondrial membrane, creating a high to low ratio. And then FADH2 and NADH, coming from our earlier Krebs cycle, are going to drop off high energy electrons to protein complexes in here. And then as electrons move across the protein complexes, they pump protons from the mitochondrial matrix into the inner membrane space, creating a proton gradient. And then when protons rush back through ATP synthase, oxidative phosphorylation occurs, producing large amounts of ATP. And then at the end, O2 as the final proton acceptor comes back with electrons and hydrogen to form water, finishing the process. And then inside the chloroplast, there are stacks of thycoids. And then these are called grana. The stacks of thycoids are called grana. And then that is surrounded by the stroma, the fluid inside of it. So light hits the pigment in photosystem 1 and 2, exciting the electrons. The electrons travel through the electron transport chain pumping protons to power ATP synthase, which is a protein complex. And then through chemiosmosis, the flow of protons make ATP and NADH, which is the energy that the plant will use for the Calvin cycle, and then will be moved on to the light-dependent reactions. Cycle which happens in the stroma of the chloroplast, and then during carbon fixation, CO2 is going to attach to the 5-carbon molecule using energy from ATP and NADPH. And in carbon reduction, energy from ATP and electrons from NADPH make G3P.